Hey guys, this is Malinki. Welcome back to my channel, Voice of Malinki. Today we will talk about life history or life cycle of Plasmodium vivax. And if you are new in my channel, please subscribe my channel. And if you like my video, please do like, comment and share my video. So Plasmodium vivax is a single-celled parasite. It is a protozoa and causes malaria. It requires two hosts to complete its life cycle. As it requires two hosts, it is called digenetic. And the two hosts are man and female Anopheles mosquito. Its life cycle includes both asexual and sexual phases. Asexual phase of its life cycle is completed in man, and sexual phase of its life cycle is completed in female Anopheles mosquito. So, this is the female Anopheles mosquito and it is carrying the parasite Plasmodium vivax. Mind it, all female mosquitoes do not carry these parasites. So, this mosquito is carrying the deadly parasite and biting a man. And we should know that a particular parasite may have different forms. So this form of parasite is called sporozoid. Sporozoid is actually a motile infective form. So when the mosquito is biting a person to have the blood meal, it injects the sporozoid stage of Plasmodium in his blood. And the sporozoids are capable of moving. And what they do? They enter the liver. In the liver cells, these sporozoids start to grow. So these are different liver cells in the liver, and the sporozoid is growing inside the liver cell. And they grow into a large and round form like this. And this form is called seasoned. Then seasoned starts to multiply by the process called multiple fission. This process is also called caesogony. Why caesogony? Because seasoned undergoes division, hence caesogony. And specifically, it is called pre-erythrocytic caesogony. Pre-erythrocytic because it occurs before entering into erythrocyte or RBC. Right? Because it is occurring outside the blood, outside the RBC, it is occurring inside the liver. That's why it is called pre-erythrocytic caesogony. Now, one seasoned forms multiple such cells inside the liver cell. These cells are called cryptozoids. Where many such cryptozoids are formed inside a liver cell, the liver cell bursts due to the pressure. Because many such cryptozoids are there inside a liver cell, and the liver cell will burst due to the pressure, and these cryptozoids get liberated from the liver cell. These cryptozoids enter into new liver cell and again grow up. Whenever a cell grows up inside the cell, it is called seasoned. Again, this seasoned undergoes multiple fission or caesogony and generates number of cells inside the liver cell, just like the previous one. Again, this is happening inside the liver cell. This time, these cells are called metacryptozoids. These cells are called metacryptozoids. Again, due to the pressure, this liver cell bursts and releases these metacryptozoites. This phase of asexual reproduction is called exoerythrocytic schizogony. Now, these metacryptozoites are of two types, micrometacryptozoites and macrometacryptozoites. Micrometacryptozoites are numerous. So, these small dots are micrometacryptozoites. These are numerous in number and smaller in size. And macrometacryptozoites are less in number. So, these bigger cells are macrometacryptozoites. They are less in number and bigger in size. Now, these macrometacryptozoites enter fresh liver cells and continue the exoerythrocytic schizogony just like the previous one. So, it will continue the same thing again and again. Now, why they do that? Because by doing that, they increase number of parasites in the body of the host. So, this is their strategy. And micrometacryptozoites will re-enter the bloodstream and invade RBC, red blood cell, or erythrocytes. That we will see now. So, the micrometacryptozoites will now enter the bloodstream and they will enter the RBC. Now, they are called as trophozoite. So, micrometacryptozoites are now called trophozoite when they are inside the RBC. And it grows up in size and called schizont. The schizont now undergoes multiple fission to form number of cells in the RBC, and these cells are called merozoites. Due to the pressure, the RBC bursts and merozoites get liberated from the cell into the blood plasma. And this phase of asexual multiplication is called erythrocytic schizogony because it is performed by erythrocytes or RBC. Now, merozoites are free in the blood plasma and they enter new erythrocytes. Then they repeat the erythrocytic schizogony. Again, they do it just to increase the cell number in the host. And some merozoites may again go from the blood to the liver cells and invade them to undergo another phase of asexual multiplication, which is called post-erythrocytic schizogony. This is called post-erythrocytic schizogony because this occurs after the RBC or erythrocytic cycle. And this cycle continues and huge number of merozoites get liberated. After that, some of the merozoites grow inside the erythrocytes. So, they grow inside the erythrocytes and they are transformed into two types of gametocytes. They are called macrogametocyte and microgametocyte. The macrogametocytes are female and the microgametocytes are male. Gametocytes remain in the human blood for several weeks but are unable to develop any further. It is necessary for them to be taken into the body of an Anopheles mosquito. If this does not happen, they degenerate and die. So this is the asexual cycle that entirely occurs in the human body. Now, sexual phase of this cycle will happen inside the body of mosquito. So, if these gametocytes which are actually inside the blood, if these gametocytes containing erythrocytes are sucked up by a female Anopheles mosquito. They reach the stomach of the mosquito where gametocytes are set free. And the microgametocytes generate flagella, and the macrogametocyte undergoes maturation process. Fusion between these two gametes generate zygote as usual. Zygote remains rounded and motionless for 24 hours. Then it elongates and becomes motile. And this zygote is called ookinete when it is motile and elongated. And the zygote gets encysted. That means a protective capsule is formed around the zygote. And the zygote is now called oocyst. Here the oocyst divides and forms sporozoites. These sporozoites get released and travel to the mosquito's salivary glands. When this mosquito bites a man, these sporozoites enter the new host and the cycle is repeated again.